The overall goal of these experiments is to induce DNA lesions using local microirradiation of cells and to observe the recruitment of proteins involved in DNA repair processes.
This method can help us to understand a very complex and profound question, such as how normal and cancer cells respond to DNA injury. The main advantage of this technique is that we are able to study the DNA damage response in individual live cells. Demonstrating the procedure will be by Soňa Legartová, a postdoc, and Jana Suchánková, a PhD student from my laboratory.
To begin, cultivate HeLa cells in DMEM supplemented with 10% FBS and appropriate antibiotics. Incubate the cells at 37 degrees Celsius and 5% CO2 in a humidified atmosphere. In a biohazard hood at room temperature, remove the culture medium and use 1x PBS to rinse the cells to eliminate all traces of serum that contains trypsin inhibitor. Add 1 milliliter of pre-warmed trypsin EDTA solution to cover the cell layer, and incubate the cells at 37 degrees Celsius for 3 to 5 minutes until the cell layer is dispersed. Add 3 milliliters of pre-warmed complete growth medium to inactivate the trypsin, and disperse the medium by gently pipetting several times. To carry out cell transfection, 48 hours before experiments, seed HeLa cells on 35mm gridded microscopy.